So the last thing I believe that we wrote on the board were the phases of the cell cycle. So I'm going to redraw that. And I don't think we said what they actually were, right? We probably named them, but we didn't write anything about them, right? Okay. So let's define what's going on in these phases. And one thing I'll say before we define them is that the cell cycle is broken up into two larger groups, aside from the four subcategories. The G1S and G2 phase, because they are not mitosis or meiosis, are sometimes collectively called interphase. Meaning this is what happens between times that the cell undergoes mitosis. And so it's the in-between phase, but it's really three different phases. So everything but mitosis falls into this interphase range. Okay, and I think that's how yours draws it, right? Yeah, it's mitosis at the bottom. Okay. And then we also have the G sub 0 phase. Okay, so let's start with G1. So the G1 phase is known as the first gap phase. And the name sort of implies it's something happening between two important phases, so it's almost like it gets, second bidding by being called a gap phase, but certainly it's an incredibly important period in the cell's life. One thing that happens during the G1 phase is that there is cellular growth. If the cell did not grow and it kept dividing anyway, it would get smaller and smaller and smaller and smaller, right? Until eventually the cells were so tiny they couldn't do anything. So we have to remember when a cell undergoes the cell cycle, it does have to grow larger, so when it splits, you'll have equal pieces in the daughter cells. So there's cellular growth. The other thing that happens, or one of the things that happens, is that the organelles reproduce doesn't feel like the right word. Well, no, that is the right word. Because the organelles have to make more of themselves, right? Because you're going to split them up between the two daughter cells. So you have growth, the organelles make more organelles, and then you have preparations for DNA replication. Which is a massive undertaking for a cell, as you might guess. To be able to copy, in the case of humans, 6 billion nucleotides into 6 billion new nucleotides. Okay, then that takes us up to the S phase. We'll circle back to the G0 phase later. The S phase is pretty straightforward. This is DNA synthesis. Outside of talking about the cell cycle, we almost always call this DNA replication. But when we're talking about the cell cycle, because it's named the S phase, we'll use the word synthesis. Why we didn't just call whoever named it didn't call it the R phase, I don't know, but that's what it is. DNA is being replicated, and so that phase, of course, is an extremely important phase because you can't undo it, right? And so when we talk about the cell cycle, I'm sure I said last time, it's like a ratchet or a clock. It only goes in one direction. So once we finish making the DNA, we can't undo that. All right, and that's going to have a consequence when we talk about cancer cells in the final installment of this unit's material. Okay, so after DNA synthesis, then we go to the, right over here, the G2 phase. And the G2 phase is a second gap phase, which again doesn't mean it's not doing something important, but it is called the gap phase. And in the second gap phase, we've already replicated our DNA, and if you're looking around the corner, you see we're about to get to the M phase, and so we'll be making preparations in the second gap phase for that. So two major things. Bless you. First is cell growth. Okay, that's a pretty major thing. And then the second one is that the cell will be making preparations for mitosis, or in the very small exception of your gamete-producing cells, meiosis. Right? We will deal with mitosis first, but we will also cover meiosis, which is the type of cell division that you undergo in gonad tissue when you're producing sperm or eggs. So it's a little bit different, as you would guess. And then the all-important M phase, which is, of course, mitosis or meiosis. On the diagram, they've just got mitosis drawn. But the way that I would suggest you think about this, because the whole thing is cell division, right? All of this is cell division. It's not just mitosis. Mitosis is not cell division. It's a component of cell division. So I would think of mitosis as nuclear division, because that's what's really taking place. You're trying to manipulate the chromosomes to split them among the future daughter cells that will be made as this process comes to a close. Okay, and so we have to be contextual here, which is that when we say nuclear division, we're not talking about, nuclear in the chemical sense, like a nuclear bomb. We're talking about nuclear as in the nucleus is divided. Okay, so those are the highlights. One last thing that we'll say before we go into some of the details is that we also have this G0 phase. And so I'll put a little box here. It doesn't blend in. The G0 phase, and that's a 0, not an O. I guess I could write that. The G0 phase is the phase where you find many of the cells in your body at this moment. Okay, not all of your cells are constantly dividing, right? Your bone marrow cells are constantly churning out new lymphocytes and other immune system cells and red blood cells. Your skin is constantly replenishing itself. Your hair follicles are producing cells that will become part of the hair shaft, and so forth. So you have a lot of what's the right word? Regeneration of tissues on an ongoing basis. But that's not true of all your cells, right? Your muscle cells are not regenerating after injury, unfortunately. Your neurons, your central nervous system does not regenerate tissues. And so there has to be a place for cells that are not preparing for DNA replication, that are not replicating DNA, that are not preparing for mitosis, that are not undergoing nuclear division. If you're not in one of those categories, where are you? Well, you're in G0. And so this is a resting state of a cell not undergoing cell division. So we have lots of cells that find themselves in this little happy place. Doesn't mean there's anything wrong, it's just what they're supposed to be doing. All right, so let's keep trucking on here. One question is, how do the cells regulate the movement between these phases? Why does a cell go from G1 to S? Why does a cell go from S to G2 or G2 to M? And that has to do with two classes of proteins. And those classes of proteins are called CDKs and cyclin-dependent kinases. Cyclin-dependent kinases are CDKs. CDKs and cyclins. So we could label this category proteins that regulate cell division. So the first of those proteins, or the first class of proteins, each of these classes has multiple members within it. But the first class are called the cyclins. And the cyclins are proteins, I'm going to use this word, proteins that cycle in abundance during cell division. Okay, this is where they got their name. At some point in cell division, a cyclin might be in very high levels, very high concentrations. At other points, the cyclins may drop down and be at very low levels. So when people discovered the cyclins, and this work was primarily done in yeast cells, because yeast cells are just like your cells, they're eukaryotes, but you can grow them in a lab, you can manipulate them, you can mutate them, you can study them, and they're just a single cell fungus. And so nobody raises any ethical issues when you manipulate yeast. But in those cells, they noticed that there was a certain type of protein that, depending on what time you measured it, G1, S, G2, or M, it would be at different levels. So they called them cyclins. Okay? They cycle in abundance during cell division, comma, and regulate CDKs. They regulate CDKs. Okay, which begs the question, what is a CDK? A CDK is an acronym, abbreviation, that stands for cyclin dependent kinases. Okay, so we have cyclins that are going up and down in abundance through the cell cycle, and then we have this group of proteins that are dependent on those cyclins that we call cyclin-dependent kinases. And when we say dependent, what we were really talking about is that the cyclin-dependent kinases work in response to direction from the cyclins, meaning they are dependent on the cyclins to know what to do. Okay, and so the cyclin-dependent kinases, well, first of all, let me ask you this. Do you remember what a kinase does? Does this make it harder to see? Is this now shining in your eyes? No? It's better? Okay. Does anyone remember what a kinase does? It's a type of protein that works in a lot of different places in the cell. So this is an example of a kinase, but there are other examples of kinases. We looked at glucokinase in the first step of the aerobic respiration pathway. That rings a bell.
It looks like enzymes.
It's an enzyme, so which means it's catalyzing the chemical reaction. Any takers? Okay. Well, cyclin-dependent kinase, like other kinases, adds a phosphate group to something, right? And glucokinase added a phosphate to glucose, and that locked the glucose into the cell. It was the first step of aerobic respiration. So in this case, these kinases are not part of respiration, they're part of cell division. But the cyclin-dependent kinases, if we add a phosphate to something, remember a phosphate is a chemical group, a phosphorus with 4 oxygens. If we add a phosphate group to something, we phosphorylate it. So a cyclin-dependent kinase is phosphorylate target proteins. Okay, so these CDKs have specific proteins in the cell that they are meant to phosphorylate. So they're targeting those proteins. Okay, phosphorylate target proteins that drive the cell through cell division. So it doesn't show that really directly on this slide, but we can maybe try and use this a little bit. So the cyclin that they're talking about here, they call it a mitotic cyclin. It has a real name, but we can just leave it at mitotic cyclin. But this cyclin, when it's at high enough levels, interacts with a CDK. All right, a CDK is dependent on the cyclin. It's a cyclin-dependent kinase. When these two guys are together, they're going to, let's see what it says. Oh no, that's not what we want. We'll talk about the checkpoint later. When those two things are together, they're going to phosphorylate targets, and that causes the cell to move from the stage of growing and preparing for mitosis into mitosis. Okay, so proteins that are phosphorylated sometimes change their activity. They become active when they weren't before, or something that was active becomes inactive. Phosphorylation is a modification that changes how a protein works. Okay, and there are millions of different examples. Okay, once this job is done, we said that the cyclins cycle in abundance, which means that once the mitotic cyclin has done its work, it's going to be degraded. So it was high, and now it's low. And once that's degraded, now this CDK is no longer doing things to drive the cell into mitosis. Why would it? It's already there. Okay, it's done its job. However, the CDKs can be thought of as tools because they are used by other cyclins to do other things too. So this CDK had a job earlier, it did its job, the mitotic cyclin exited stage left, and now this CDK is free to do work for someone else. Well, that someone else is a G1 cyclin. And so when these two things interact, the cell moves into G1. Okay? And when G1 is done, that cyclin is no longer needed, and so it gets degraded. So these cyclins have these passing transient roles in the cell cycle. Once they do their job, and once they tell CDKs what to do, then they disappear until the cell is ready for them to act again. Okay, so this, pardon the term, this is a endless cycle, literally. This is what's really going on. Don't learn this slide. How could you? But if you're looking at this and thinking, gosh, that's simple, or maybe you're not thinking that, but when I look at that, I think, that be it? That's what makes our cell go through cycles? And it's not, it's just part of it. What really happens is much more complicated. So here are two CDKs that are driving the cell from G1 into S phase. These CDKs are activated by certain cyclins, cyclin D and E. Those things are interacting with all of these other proteins. So everything drawn here as a circle is a protein. A protein might be activating something. If it is, it'll have an arrow. So CDC25 is activating this. If it's inhibiting it, it will have this little blocked arrow. So people have mapped out all of these interacting proteins in the cell that regulate the cell cycle. We're just looking at a glimpse, just a glimpse of it. So I don't want you to walk away thinking it is that simple. It's not. We're not going to learn this in this class. That's okay. It's not our purpose. But I just want you to know that the layer of complexity is pretty deep, which is fascinating. I think it's beautiful. Okay. Now, one question that should come up is, why is the cell dividing in the first place? Right? If CDKs and cyclins are there, why are they there? How did they get there? Why are they going up and down in abundance? And the answer is that the cell is sending a signal to tell the nucleus to make RNAs that will become proteins that will then make the cell divide. Okay, and so this process, again, is more complicated than the one we're going to go through. This is what's really happening, and this is just a small version of it. Okay, so let's take a look at this for a minute. Let me make some space. If you're following along on the review sheet and kind of monitoring where we're going, this was right next to apoptosis. So it said apoptosis, and then I think it said cell signaling or cell transduction pathway. There were two little bullets. I didn't bring my sheet to reference, but there were two little bullet lines before we got to mitosis. This is the second one. It's kind of embedded into PowerPoint. Okay, so this is called a signal transduction pathway. Right? To transduce a signal means you're sending it from one place to the next to the next. So a signal transduction pathway takes a signal from one part of the cell and moves it along a chain of characters until it gets to another part. And so this particular signal transduction pathway is sometimes called the MAP kinase cascade. So that's a part of this pathway. Actually, it's kind of a subcomponent of the pathway. But if you hear MAP kinase cascade, it's all connected. Okay, it's all part of it. So in the signal transduction pathway, the trick is to have something outside of the cell interact with the cell, and then that message can be carried to the nucleus. And once it's in the nucleus, RNAs can be made that can then produce proteins that are involved with cell division. So the question is how to get from point A to point like M, really. We go through quite a number of points to get there. And so let's start at the outside. And so the process begins with a ligand. Remember, a ligand is anything that interacts with a receptor. So there was a ligand that interacted with the death receptor to cause apoptosis. There are ligands that interact with receptors to make melanin and pigments, bless you. There are ligands that interact with receptors to release insulin. There are ligands that interact with receptors to absorb glucose. This is just a way that the cell tells, or a way that cells tell other cells what to do. So a ligand interacts with a growth, they call it a growth factor receptor on that. Well, yes, the GF of EGF is growth factor. So a ligand interacts with a growth factor receptor. Okay, and this is going to trigger an event on the inside of the cell. By interacting with something on the outside, it's going to trigger a change on the inside. And that change is going to be the phosphorylation of the intracellular part of this receptor. The receptor spans the membrane, right? It's a transmembrane protein. So when the ligand and the receptor interact, there's a change. So the second step is that proteins, they call them relay proteins. Often they're called adapter proteins. We'll say proteins associated with the receptor, and I'll put relay proteins, so if you look at this diagram later, you'll know what I'm referring to. These proteins activate RAS, R-A-S. Okay, in the diagram, RAS is one of these aqua colored molecules. If I can achieve this. Can I do it? Yes, I can. Here's RAS. And so in order to activate RAS, we have to change something about it. Because RAS is like the first big switch, the first big domino to fall that triggers this chain reaction getting to the nucleus. So it's an extremely important protein. When RAS malfunctions, bad things happen. So if you have a cancer cell growing inside a body, an organism, oftentimes RAS is not working. Because if RAS causes cell division, that is cancer, right? Cancer is uncontrolled cell division. So if we fail to regulate this, cancer is a huge risk. We'll circle back to that later. Okay, so to turn RAS on, something's going to happen. So to activate RAS, what we have to do is we have to change RAS's association with a molecule that I don't think we've talked about it yet, but we talked about something that will give you context for it. So GDP is very similar in structure and function to ATP, which is ATP minus a phosphate, right? Remember, when ATP gives away energy, it loses a phosphate, it becomes ADP, triphosphate to diphosphate. This is the same type of molecule, only here we're not really using it as an energy molecule. GTP and GDP play a different role in the cell. Sometimes they are energy molecules, but in this case, they sometimes play a role in the cell where they alter whether a protein is active or not. But I tell you all that because I don't want you to look at that and just think, oh, great, another molecule to learn. This is just like ATP, so we should be somewhat familiar with this already. So RAS GDP, in order to be activated, we need to exchange that GDP with a GTP. Now, if I were in charge of life, I might have engineered this so that we just add a phosphate, right? That's the difference, diphosphate to triphosphate. Seems like the easiest path. Let's just phosphorylate it and we're off to the races. It turns out that is not how it happens in real life. In real life, what happens is the GDP leaves and the GTP comes in and replaces it. So I wouldn't have engineered it that way, but nobody asked when life was evolving how I would do it. But that's a little unexpected to me. I've always thought that was a little strange. In any case, now RAS is active. And so we could say on this side, let's use red. Color of blood. So we go from the inactive state to the active state. And this happened because a receptor was activated by binding to a ligand. Okay. All right, so now we have an active RAS. So what good does that do us? So much good. All right, what step are we on? 3. All right, so now we have an active RAS. If you notice, RAS is going to interact now with one protein. Oh, excuse me. Welcome to 8th grade. One protein. This protein is going to activate with the second protein, or interact, excuse me. That one's going to interact with the third protein. So we have 3 proteins, RAF, MEK, and ERK, that are now going to get involved. This is the MAP kinase cascade. So RAS, GTP, which I'll put in parentheses, the active form, let's see, activates the MAP kinase cascade. And I don't want to break my own heart, so I'm not going to do a survey of how many of you intend to pursue a biology or biomedical or vet major, because I'm afraid I might learn that I scared you away from this beautiful discipline. I hope I have. There's room for everyone under our science umbrella, even those with broken souls. In fact, most of us under the science umbrella have broken souls. It's the process. But if you do stay in this major and pursue a degree in science, which I hope you will, if that's something you're interested in, you're going to see the MAP kinase cascade a lot. So this isn't a passing topic that you're going to learn once. It's important. You're going to see it multiple times. You'll see it a lot in genetics. You'll see it in cell biology. You'll see it in biochemistry. You'll see it in neurobiology if you take that. You'll see it in developmental biology if you take that. It's going to recur microbiology. Recur and recur and recur. So it's really important. So what the MAP kinase cascade does, cascade is usually used to refer to like a flowing thing, right? Like water cascading down a waterfall. And so in this idea, this expression of that word, they're saying one thing activates the next, that activates the next, that activates the next. It's like this flow of information and activation down multiple proteins. And so the first of those proteins is named RAF. So the MAP kinase cascade first activates RAF, which then activates MEK. I know they're weird words, which then activates ERK. Wish I could remember what all those were named after. It escapes me now. They're named after strange things. But in any case, RAF, MEK, and ERK, it's hard, well, maybe it says here. RAS activates RAF, which starts a protein kinase cascade in which RAF phosphorylates MEK, and then MEK phosphorylates ERK. ERK enters the nucleus and phosphorylates transcription factors. So one phosphorylates the next, phosphorylates the next, phosphorylates the next. So each kinase phosphorylates the next. in the series. That's the cascade. All right, and when RAF phosphorylates MEC, phosphorylates ERK, when it gets to the last one, ERK, let's put that as bullet point 4. ERK activates proteins in the nucleus Okay, ERK is the only of the 3 MAP kinases that can go into the nucleus. The others stay in the cytosol. But ERK, when it's activated, it's like a messenger that is sent into the nucleus to start some activity to create more proteins. So ERK activates proteins in the nucleus that lead to cell division. Okay, it activates proteins in the nucleus that eventually lead to cell division. Now, we haven't talked about what a transcription factor is. We haven't talked about messenger RNA being translated to proteins. That's going to be our last unit. So I'm not going to assume you know or remember that from previous classes. Maybe you do. Big topic, but I don't know what you know. So let's not worry about that yet. It's not part of this unit. So we'll keep it simple and say it leads to activation, or it leads to eventually cell division. Okay, so that message came from outside, took a lot of steps, got to the nucleus, and now cell division can occur. So every time a cell jumps into G1 phase and goes to S phase and so on, that's because this process has initiated the production of proteins. Can anyone guess what these what some of these proteins might be? Cyclins, you say? Probably. CDKs, I hear? Yeah, probably. So those are things that will drive cell division. Okay? All right, so there'll certainly be some questions about that on the little thing I call your exam. All right, and again, much more complicated than I'm telling you, but what I'm telling you is not untrue, it's just part of the story. So many more beautiful, cool things happening. All right, now, leading into mitosis, now let's get down to some of the nuts and bolts of cell division. This is probably going to circle back to things you've seen in previous biology courses or science courses in your scientific upbringing. But one of the confusing and confounding parts of cell division is how we describe and talk about chromosomes. And the terminology can be irritating sometimes, but we have to learn it. So each chromosome replicates prior to mitosis. Remember that mitosis is happening down here, but DNA synthesis happens 2 stages prior. So each chromosome replicates prior to mitosis. At the start of mitosis, the chromosomes become compact. So normally we wouldn't see DNA looking like this in the cell. If we took most of your cells and stained them and put them under a microscope, all we would see is that the nucleus had some dark staining. We wouldn't be able to tell that there were chromosomes in there. But if we found one of your cells that was undergoing mitosis, we probably would be able to see chromosomes that look like this X structure, okay? And that X structure contains 2 replicates of the original chromosome, all right? And those 2 replicates are called sister chromatids. Now, here's where some of the terminology gets a little weird. Also, this diagram is not great. We're doing the best we can. So these should be connected. I'll try and color it in. There we go. Whichever software engineers wrote the coding for how the mouse will translate to drawing on the screen, they must have said, let's make it like a 2-year-old, no matter how good they are. Okay. There we go. So all that should be connected. They're not split yet. All right, so the terminology that we want to be comfortable with is if we see something like this, right, which is a single chromosome prior to replication, this is one chromosome. Did you do the cell division lab yet, where you look through microscopes and count cells that are in anaphase, metaphase? Does this ring a bell? Okay, good. You will, and this will give you some, hopefully, some preparation for that. That's a good lab. Actually, I like that lab. All right, so we have one chromosome. So if we go through DNA replication, we then will have a structure like that. That is not two chromosomes. Even though we've replicated one chromosome, if someone said, I'm giving you this pie, and you say, okay, I'll replicate that pie, now I have two pies, that's not the same as this. When you replicate a chromosome, you don't have two chromosomes. You think you would have two chromosomes, but you actually have one chromosome. Extremely unsettling terminology, in my opinion. But that one chromosome has two sister chromatids. In the world of mitosis and meiosis, you don't become a chromosome until you have split from your sister chromatid. All right, so right now, it is still one chromosome. All right, if we undergo mitosis, and specifically the anaphase phase where the chromatids split, then we have two chromosomes. But not until then. Let's take a moment to distribute this a little bit. It's turning the hot room into a convection oven. Distributing the hot air so everything gets burned. Hello, I am your father. Maybe I could strap it to me with one of those baby carriers. Walk around. Or then you just get in my body. That's not gonna help. It's helping a little. If I could tilt it up, I could have the Carrie Underwood effect. Air flowing as I talk. Okay. All right, what do we want to say? Oh, we have some terms. So within the chromosome, we have a few things to label here, the kinetochore and the centromere. Let's make sure we understand what those are, because they're often confused for each other, and they're definitely different. And so on a chromosome, there is a region called the centromere. Let's see how they define it here. The region of DNA beneath the kinetochore. Bless you. Did I stir up all the dust by spraying you with that fan? Sorry. The centromere, we can add to our list of aggravations. Extreme heat, mold, dripping ceilings, angry professors. One's on edge because of the heat. Try and keep that into account. So the centromere is exactly that. So a mistake when people look at a chromosome sometimes is they see this structure and they call this structure the centromere, but the centromere is the DNA. It's not the stuff stuck to the DNA, it's the DNA itself. That's where we want to really draw the line. So the centromere is a DNA region that is bound by the kinetochore proteins. Okay, so if we took the kinetochore proteins away from this condensed chromosome, there would still be a centromere. It's part of the chromosome, it's part of the DNA. The kinetochore, on the other hand, kinetochore is a complex of proteins. I don't know if we have that named there. Now let's look at this one instead. So the kinetochore is a complex of proteins that binds the centromere. It's not just one protein as it often appears in drawings. It's a complex. I think it's in most organisms it's like 20 or more proteins. It's a big group. But it binds to the DNA at the centromere. Okay? The kinetochore is going to serve an important role later because the microtubules that move the chromosomes around need something to hold on to. They don't just grab the DNA, they grab the kinetochore. The kinetochore is going to serve kind of as the handle that the microtubules are going to use to move chromosomes. Maybe we should write, so a complex of proteins that bind the centromere. The microtubules will attach to the kinetochore. To the kinetochore. Cheers to whoever just popped the top on their cold drink, formerly cold drink. I don't know how you waited this long, unless you're on like the third one. Okay. Anything else about this? I think we're good. All right, let's define some of the structures associated with cell division, and then I believe we'll jump right in, if I'm not mistaken. Yeah. Okay, good. So some of the structures that we want to understand, we've already seen the kinetochore and we've seen the sister chromatids. And in this diagram, we're looking at 2 chromosomes, right? 2 chromosomes, but 4 sister chromatids. And the kinetochores are bound to the DNA at the point that we call the centromere, all right? And then the microtubules are going to attach to the kinetochores. So these microtubules grow out and attach to those chromosomes at the point where those protein complexes are bound, right? They can't bind the DNA directly, so they bind the kinetochore, which in turn is bound to the DNA. And you notice that these microtubules are growing out from a region called the centrosome. Okay, so make sure you feel comfortable with this diagram and what it's demonstrating. So the microtubules come in more than one form. So let me write a couple of these things down in our terms. So if you were organizing your notes the way I would, we had centromere, kinetochore, those terms, and then the next terms I would put would be centrosome. The centrosome is the origin of microtubules. Okay, so the microtubules are originating at this structure called the centrosome. It says it has a centriole pair. If you ever look at a really high resolution electron microscopy image of a cell undergoing division, you'll see the centrioles and they look like cheese combos. Exactly like cheese combos. It's almost unnerving and appetizing at the same time. But that's what they look like, little tiny cylindrical cheese combos. There's no better way to describe it. And so those centrosomes are the source of the microtubules. They grow out from there. Okay, the microtubules don't stretch, but when they get longer, they're actually they're polymers. So you have lots of little subunits that add on. It's like, I wish there were a different representation of it. It's as though this was like a long chain of little Lego blocks. You just add one more each time to make it longer, and then you take blocks off to make it shorter. So it's not that they're growing and stretching, you're actually building one thing onto the next to make it the length you want. Now, the microtubules are indeed moving the chromosomes around, but if you notice, there are also these things called polar microtubules. Okay, so let's write this down. So the origin of the microtubules, so we have, oh man, spelling challenge. We have the kinetochore microtubules that move the chromosomes. Okay, those are the ones we're going to focus on, but we do have a second category that we'll mention because they matter. Those are called the polar microtubules. And the polar microtubules are not attached to the kinetochores. Instead, they're attached to each other. It's not really shown very well here. It just kind of looks like they're near each other. But what's actually happening, get on my toddler pen, these guys are actually attached to each other. It's too hard to draw behind that. And so the fact that these things are attached to each other means that, well, let me ask you, if they're attached to each other like that, and one of the polar microtubules or both of the polar microtubules get longer, what will happen to the cell? So our cell is like this. The polar microtubules are running from one side and running from the other, and they're attached at the middle. What will happen to our cell if the polar microtubules now get longer? It extends, right? And that has to happen. Our cell originally was like this, but we want it to get bigger and bigger and elongate it, and then it'll split into two. So the polar microtubules are not in the business of moving chromosomes around. They're in the business of making the cell larger, okay? So they matter. Okay, now back to maybe some familiar territory. Phases of the cell division nuclear separation moment, mitosis. All right, so interphase precedes this. Remember, interphase in our diagram is everything other than mitosis. So the DNA has already replicated in interphase. Our organelles have reproduced to make more organelles. The cell has grown, started to elongate a little bit, and now we hit nuclear division, which is mitosis. So oftentimes this is written as 4 phases: prophase, metaphase, anaphase, telophase. This book and some other books, just depends which reference you use, throw in an in-betweener phase called prometaphase, which is fine. We'll follow your book's lead. But prior to using this book, I'd never heard of prometaphase. We skipped right over that. But since this book uses it, we'll go for it. So the stages of mitosis. The first one, let's hit the highlights of each stage. And you'll want to be sure that you feel comfortable recognizing them. Let's say you saw a picture of it, which you will in the lab. That'll be a big part of that cell division lab. Let's say you saw a picture of it on exam, or you saw a description of it. This was happening. You'll want to say, oh, that must be metaphase. That must be anaphase. That must be prophase. All right, so in prophase, you can see the main difference in the nucleus is that now the chromosomes start to look like little wormy things, whereas before it was just a blur. This is how our cells look most of the time if we were able to stain the DNA. Rarely would you be able to pick out chromosomes within that. Okay, so in prophase, the chromosomes condense. Okay, the nuclear envelope, which remember, the membrane around the nucleus is a double bilayer. So 4 layers of lipids. So oftentimes instead of calling it a membrane, people call it a nuclear envelope. But it is a membrane, a double membrane. So the chromosomes condense. What's that?
Oh, it got this heating up.
Oh, okay.
I figured it was hot in there.
I wonder if we can put it on the other side. Thanks, Dave.
Right there on the wall there, right there.
Let's see. You want to blow?
Need one right there behind you.
Oh. I might be
Sorry,
able.
Bobby, I figured it was hot in there.
No, I appreciate it, Dave. Thanks. Might be able to achieve my Carrie Underwood moment after all. Tried to go to 3. She has some dark songs. I like the one where she's in the cellar contemplating killing someone. Hard to beat Carrie Underwood. Okay, so the chromosomes condense. The nuclear envelope starts to break down. Now, if you were not going to include prometaphase in your listing of stages, you would say the nuclear envelope breaks down. But one of the hallmarks of prometaphase, if you do include it, is that the point that defines it is that the nuclear envelope is completely gone and the microtubules have reached out and attached. And so oftentimes you might include that in this phase. So just FYI, because you may see it differently in the future, but that's how we'll do it. And then so maybe I can tilt it a little. Get some elevation there. Next level around here. All right, so that's happening. Then we'll have prometaphase. We'll include it here. And so in prometaphase, I'll abbreviate, nuclear envelope is gone. GBNF, gone but not forgotten. It'll be back. Nuclear envelope is gone. Microtubules attached, or let's see, I'll say attached. Microtubules attached to chromosomes. Now, I'm implying that if they're attached to the chromosomes, it's to the kinetochores of the chromosomes. I'm going to save just a little wordage by leaving that out. Okay, anything else? No, that's okay. All right, so now we reach metaphase. This is probably the most recognizable phase of the mitotic phases, because those chromosomes get manipulated to be positioned across the middle of the cell. So visibly, it's a pretty striking phase, so this is going to be 3 on the list. Okay, so in metaphase, the chromosomes, remember, they still have their sister chromatid with them, and so this just says it that way, sister chromatids. I prefer to say chromosomes. You can say it either way. They're both accurate. But the chromosomes are all lined up along the middle of the cell. So this goes by different names. Sometimes people call it the metaphase plate. That's what your book calls it. So I'll put chromosomes line up at metaphase plate. You might have learned it as the equator. Some people call it the equator or the midpoint. Or sometimes people call it midline of the cell. Okay, but either way, whichever of those terms you use, they're all pretty descriptive and intuitive. It means right across the middle. Okay, so it's a remarkable thing, but these microtubules are able to grow and contract and grow and contract until those chromosomes find themselves organized in this nice row across the middle of the cell. And the point of this all is going to be to divide everything equally. So now you're lining everyone up and you're going to split the sister chromatids apart. And so in the 4th phase, anaphase, this is when the microtubules, if they were to start shortening, right, if they were to start removing subunits and getting shorter, the sister chromatids are going to have to make a choice. And that choice is going to be to say goodbye to their sister and become a chromosome in their own right. And so when the kinetochores split and the centromeres split apart, the chromosomes, formerly chromatids, are now all grown up. So we'll say that the sister chromatids are separated to become chromosomes. And I don't know if this one doesn't say it. A lot of times the phrase that people use for this, which I think is both funny and appropriate, is they say they become chromosomes in their own right. Like they're no longer just half of a chromosome, and now become chromosomes in their own right. I always thought that was kind of funny. All right, and again, look at our polar microtubules, always in the shadows. But there they are. So they're pushing apart and the cell is getting bigger and bigger. And so anaphase, the chromatids separate to become chromosomes. Then, number 5. Our last phase is telophase. They've kind of clumped cytokinesis in there with it. But we're going to separate them. So telophase, the nuclear envelope, which has been disassembled, right? But it's still there, just the pieces are just not together. So the nuclear envelope reforms. Okay, back here, it had begun to dissociate into vesicles, so it's being packed away for later, and now it's back. And so the nuclear envelope reforms, not reforms, but reforms. Okay. And the cell starts to separate into 2 cells. And I'm going to put that as a separate stage. You can put them together if you want, but I think of it as being distinct. So cytokinesis is when everything is ready to be 2 individual cells, and so the only thing that's left is to make sure the plasma membrane separates, so there's no longer any, connection between the 2. Okay, so in cytokinesis, the 2 daughter cells form, and depending on whether you are an animal or a plant, one of 2 things is going to happen. If you're an animal, you are going to form what's called a cleavage furrow, and actually, there we go. And the cleavage furrow is, well, just what it sounds like. To cleave means to separate. A furrow is like a little valley in something, like a furrow in a farm field of the farm is where you create a little valley and you put your seeds and cover. That's what we're talking about here too. You have that little valley, which is going to eventually be cut between the cells. So cleavage furrow. And the cleavage furrow forms in a pretty neat way. You have proteins inside the cell that make a ring. And if that ring gets smaller and smaller and smaller, imagine if you had, and actually some people make bread this way, they make a little ball of dough and they put a piece of string around it like the size of a piece of yarn, or the diameter of a yarn, and they tie it in a knot and they pull the knot, and when they pull it, the 2 pieces of dough split into nice little balls. That's what's happening during mitosis. Maybe that bread making was inspired by cytokinesis, for all I know. But the formation of the cleavage furrow and then that contractile ring gets smaller and smaller until eventually they divide. On the other hand, if a plant cell is dividing into 2 daughter cells, they can't do that. They can't do that because there's a cell wall. And so inside the plant cell, you have to account for first the splitting of the plasma membranes and then the formation of the cell wall. And so what they do, once everything is intact and in its place, the nuclear envelope you can see has reformed a structure called a cell plate, which becomes the future cell wall, starts to be built between them. Okay, so that will separate it as opposed to a cleavage furrow. Okay? And then eventually you've got your 2 cells. All right, now let's take a moment to think about something that I maybe should have led with. Got a few minutes left. Your internal temperature is probably reaching around 375, so you'll be done soon. When we first started this, what I probably should have said is the difference between mitosis and meiosis, other than just saying one occurs to produce egg and sperm and one occurs in all your other cells. So let's clean that up now. So mitosis first of all, occurs in somatic cells. Somatic being another term for body cells, as people call it, which is very vague, but what it means is cells that are not gamete producing cells. They're not going to make sperm or egg, and so we call them body cells, but a more technical term would be somatic cells. Okay, so that's mitosis, and the purpose of mitosis is to produce, or I'll say more grammatically, it produces two diploid cells from a parent diploid cell. Or I guess to follow the terminology, as you say, two diploid daughter cells from a parent diploid cell, single. Clean that up. Heat's getting to my brain. Okay, and we haven't spoken a lot about diploid and haploid, but we will need to confront that now. Diploid means two copies of each chromosome. Now, are there exceptions to this? Of course there are. In the body, we have cells that have multiple nuclei. So you have some multinucleated cells which have more than two copies of each chromosome that are undergoing mitosis. You have cells that have no copies of the chromosome, right? Your red blood cells get rid of their nucleus during their maturation process. So they're hanging out there with no chromosomes at all. So yes, obviously there are exceptions, but this is the most general rule we can say. Now let's contrast that with meiosis. In meiosis, this occurs in germline cells and the purpose of meiosis is different than the purpose of mitosis. Germline cells are gamete producing cells, and so in meiosis we produce 4 haploid gametes, and gametes, remember, are egg or sperm, depending on the parent, from a diploid parent cell. Right, 4 haploid gametes from a parent diploid cell. And so a big difference between meiosis and mitosis, other than the obvious that I've just said, is that it goes through division twice. Okay, it separates the cells into 2 daughter cells and then 2 more daughter cells. So you end up with 4 products instead of 2. And the 4 products, if you started with a single pair of chromosomes, the products of those are going to be a single chromosome each. So in the case of humans, we start out with 46 total chromosomes and we'll end up with 23. All right, let me think a second. Okay, let's make this our good stopping point because I just can't bear it anymore.